So in this presentation, I'll describe the different steps that you can follow when you conduct an individual classification analysis using neuroimages. So as you've seen from the previous talks, the analysis pipelines that are used for individual patient classification or regression can vary, but they often include 4 elements. The first one is the training dataset, which has to have a sufficient number of subjects and with well-characterized clinical properties to reliably express, so really the disease effect against the noise that you can have because of intersubject variability. Then you have the feature extraction and optionally like the dimensionality reduction elements. Here, basically, the acquired images, they usually need to be pre-processed to extract meaningful features. So, for example, from anatomical MR image, you can study the gray matter density in each voxel of the image or the cortical thickness in different regions of the brain. Then you have the model training and optimization. And so at this step, you select a supervised algorithm, such as an SVM or random forest. And the parameters of the models are optimized so to maximally discriminate one group from another, so based again on your training data sets. And this is done usually using cross-validation. Finally, you can apply the model to the test data that in pre-process is the same way as the training data sets. So if we take Alzheimer's disease as a use case, this type of pipeline can be used to perform classification tasks, such as clinical diagnosis classification, like for example, differentiating cognitively normal subjects from patients with Alzheimer's disease or Alzheimer's dementia. You can also perform some more like, let's say, predictive tasks of the evolution from the prodromal stage of the disease to really the, for example, Alzheimer's disease. But this pipeline can also be used to perform regression tasks, for example, to predict the time of onset or future clinical score. So this is like the theory that you've seen, but when you're about to make things more concrete, maybe like a few questions will arise. The first one has to do with the training and testing data sets, because if in your institution you're not lucky and you don't have like a data set, then you want to know actually where to find all this data. Then you should wonder about what is the best way to actually organize your data, because you see that it's not always done in the best and optimal way. Now, regarding the feature extraction block, you can actually wonder what type of features you can extract from your images. Once you've selected that, you are ready to start playing with like machine learning. And here again, you have a few choices to make, which are, for example, which are the tasks that may be of interest, which classifier you can choose, which cross-validation strategy you might want to implement. And finally, maybe more importantly, you would like to know the influence of all of these choices on the classification performances. And so in this presentation, I'll try to give answers to these questions. And the tool that I use to help answer several of these questions is called Clinica. So Clinica is an open source software platform for reproducible clinical neuroimaging studies, so which is written in Python and relies a lot on NiPipe for pipelining. It can take as input several modalities such as MRI and PET, and then it provides complex processing pipelines that involve a combination of different software packages such as SPM or FreeSurfer. Once you've extracted your features from the data, they can be used as inputs of statistics or machine learning analysis approaches that rely on packages such as scikit-learn. The input data are organized following the brain imaging data structure or bits format. I will come back to that later. And the output follows the Clinica process structure or CAPS format, which basically shares like bits philosophy. And because not all datasets are organized in bits, Clinica also provides tools to convert publicly available datasets to bits. And again, I will come back to that later. If you want to know more about Clinica, we have several posters during the conference, and we also have a talk in the Open Science Room on Thursday. So our first question, where do I find data? Well, it might be an obvious one to some of you, I hope most of you, but you can use actually public data sets. And there are like many public data sets. So for example, for dementia, you have like these three that are probably the most famous, but you have also data set for other conditions. And maybe a first website that you can check is the Lonely website, where you have like all these data sets. So not all of them are open to the public, but for most of them, you just fill a form and then you can have access to the data. So going into a bit more details, first you have the OASIS, so open access series of imaging studies, that is actually composed of like 3 data sets. The first one is consists of basically cross-sectional images, T1 images for like more than 400 subjects. Among these subjects, around like 100 were diagnosed with mild to moderate AD. OASIS 2 is a longitudinal data set of 150 subjects, again with like T1 images. And again, you have some controls, but also some subjects with various stages of AD. And you also have like OASIS 3, which is a retrospective compilation of data for more than 1,000 participants. Here you have not only like anatomical MR, but also other types of MR and some PET data. Then you have the ADNI dataset, which is also like very famous. So ADNI is an ongoing longitudinal and multi-center study that was launched in 2003. The primary goal of ADNI has been to test whether serial MR, PET, and other biological markers, but also clinical and neuropsychological assessments can be combined to measure the progression of mild cognitive impairment, or MCI, and early AD. So in ADNI, you have more than 1,500 participants that were recruited across like North America, and you have like actually 4 phases now: ADNI 1, GO, 2, and now 3. And again, you have patients with AD, with mild cognitive impairment, some controls. And depending on the time points, you have some MR images, some PETs, and also some other, like, for example, blood biomarkers and other things. So really a lot of data here. And then you have, really in short, the Australian version of ADNI. Again, like 1,000 participants with AD, MCI, and some healthy controls. ABLE follows like the ADNI 1 protocol. So you have like MR and PETs, but also biomarkers, some genotype, and other type of assessments. So I think that's already quite a lot of data. And let's say that you pick, for example, ADNI, and you download the data set. If you check what you're going to end up with, it's something like that. So you have like the name of your subjects, and then some more or less cryptic folder names. You can recognize some name of sequences, but it's not really obvious. And then in blue, you have like sessions. And finally, like your images in DICOM. But this is not really what you would like to work with. Something like that would look a bit better. Same like the name of your subjects, the session clearly identified. The modalities also clearly identified. You have like Nifty files for the images. You also have some metadata that are associated to that. And this is actually what you have when you follow the BIDS format. And adopting actually a common standard to describe your data and to organize your data on your disk can provide multiple benefits. It minimizes like regression, reduces the chances of errors. It allows you to optimize the use of your data software packages, and it allows you to also verify the consistency and the completeness of your data sets. So there are several steps to follow if you want to convert a dataset to bits. The first one is to convert your DICOM images to Nifty. You can do that using converters such as DCM to NIX, for example. Then you have to create like the folder structures, rename and copy like the Nifty file in it. Then you can add some like remaining data, for example, details of the experimental paradigm for your task fMRI data. You can also add some missing like metadata, so that are this is done using like JSON files. And finally, you can validate your data sets. So this can look like a lot of work, especially for like the ADNI data sets. And that's why actually in Clinica we provide tools to do that automatically. So you just basically have to download your data set, well, of course, install Clinica, and then it's just like one command line. It's basically Clinica convert any 2 bits, the folder where you downloaded the data and the output folder, and that's the only thing you have to do to have all your ANI data sets nicely organized, but also we do some data curation. So for example, if you have like several MR images for like just one session, then it's going to pick the one with the highest quality based on some like CSV file that ANI provides. Okay, so now that you've found some data, you've organized them, you can actually start looking at them. So if we take here like an example of male AD subject age like 74 from the ANI2 datasets, you can look at his anatomical T1 image to know a little bit about atrophy. You can also look at diffusion MR data to know about potential white matter alterations. With FDG PET, you can know if there's some hypometabolism. And finally, with the amyloid PET, you can know if actually the amyloid protein is present in the brain in a large quantity. But these are actually quite raw data, and it's not really what you're going to feed to your classifier. Before, you need to extract meaningful features from the data. There are several types of features. You can first look at voxel features. And here, basically, your subject is seen as a collection of values at each voxel of the image. So you can typically do that when you look at like gray matter maps, for example, or PET SUVR maps. So SUVRs is just a way to intensity normalize your PET images. Here, if you want to work with a lot of subjects, you will first have to perform inter-subject registration into a common space, so to have a voxel-wise correspondence across all of your subjects. You might want to look at surfaces, and in this case, you have vertex-based features. Here, each subject is seen as a collection of values at each vertex of the surface. This is typically used, for example, for cortical thickness maps, but you can also look at PET SUVR maps that you can project on the cortical surface. Here again, you need to perform some inter-subject registration to have everybody in a common space and to have a vertex-wise correspondence. You can also look at regional features. Here, so each subject is seen as a collection of values for each region of the brain as defined by the atlas that you will pick. Here, you can look, for example, just at the volume of each region. You can look at the average, for example, thickness within a region, or the average fractional anisotropy, again, within a region. You have like many types of atlases that you can check on the website there. Basically, you can divide them into like really some based on the anatomy and some based on the function of the brain. Then you might also want to look at like graph-based features using the graph directly, so for example using graph kernels, but also using indices that you derive from the networks and that characterize the global and local topology of your graph. If you're not very familiar with all these processes, you can here again use like Clinica to basically perform all these pre-processing steps to extract your features. So we have pipeline to extract features from T1 MR images, diffusion images, and PET. From that, you can extract voxels, vertices, or regions. And again, it's just like one command line, basically, where you pick like the pre-processing you want. You specify like the input, the output folders, and that's pretty much it. You don't have to tune any parameters because we've made these choices for you, and we pick like the parameters that we found to be optimal. Okay, so now that you've pre-processed all your data, then you might want to know which task you can actually perform and which can be of interest. So if we still keep like the example of ANI, in ANI you can have like several groups based on their like diagnosis. So the most obvious one maybe are like the cognitively normal CN AD, and MCI, so mild cognitive impairment. And these groups, we usually make them based on the diagnosis of the subjects at baseline. But we can also look at a bit more like sophisticated groups, for example, with the SMCI or PMCI, so which means like stable MCI subjects or progressive MCI subjects. And basically, we define these groups are being subjects that were diagnosed as MCIs at baseline, were followed during at least 36 months, and convert to AD for like the progressive, or did not progress to AD for the stable during this time frame. So from all of these data, so the task that you can select, actually, you could maybe try like all the combination, but it wouldn't make much sense. So, for example, you can look at so the controlled versus the Alzheimer's disease, or the equivalent, but a bit more like difficult, controlled versus progressive MCI. And here, basically, you perform like a clinical diagnosis classification task. But you can also look at, for example, stable versus progressive MCI, while here it's some sort of like predictive task of the evolution of the subjects. Then you can pick your classifier and the cross-validation that you want. So in terms of classifier, so I'm not going to go into details, we've seen that in the previous talks, but basically you can pick like a linear SVM, some logical regression, some random forest. And then in terms of like the cross-validation, I think we've seen from like Pradiv that we have a few like options. We like privilege like the repeated holdout with like many repetitions that we've seen before. And we optimize like the hyperparameters of our classifiers using nested cross-validation. Yeah, I think we've seen like a lot of that this morning, so I won't go into the details. But basically, once you've done all that, you end up with like some possible inputs. Let's say in terms of imaging modalities, from like ADNI, we have like the T1 weighted ones, and we want also to study like FDG PETs. And in terms of features, we are going to look at voxels, but also at regions. Then, for example, here we pick like 3 classifiers, but we want to like test and compare. And we also implemented like 3 types of cross-validation. And again, like with all of that, we will be able to see like the influence of like the choices that you can make on the classification performance. So in the following, if I don't mention anything, the classifier will be like the linear SVM, and the cross-validation is the repeated holdout. So we compare like the balanced accuracies obtained for voxel and regional features that were extracted from both like T1 and FDG PET images, so from ADNI participants, and so here using a linear SVM classifier. So basically you can see from the results that there is really no notable differences between the mean balance accuracies obtained using voxels and regional features. So in our case, basically the influence of the type of features, there's nothing really systematic about it. So in this case, like the choice that you make between voxels and regions doesn't really have a huge influence on the result that you'll get. Then you might want to know like the influence of the classification methods on the results. So here we perform regional regional-based experiments, so regional features using regional features for 3 classifiers. So for now, it's just like using features extracted from T1 MR images. And basically, the results show that both the linear SVM and logistic regression with the L2 regularization models, they lead to a similar balanced accuracy, which is higher actually than the one obtained with the random forest for the different tasks that we tested. So yes, we tested control versus AD, control versus progressive MCI, and stable versus progressive MCI. And we basically see the same thing when we extract the features from the FDG PET images. And we think that the underperformance of the random forests might be caused by the nature of the brain imaging data that contains basically relatively homogeneous values. and which should show a dependence across voxels or brain regions. And these characteristics of the data also could explain why techniques that try to find a smooth combination of features, so such as the one using the L2 regularization, are more suited for single modality classification problems. But on the other hand, the random forest or other like ensemble method, they could be useful when combining features from different modalities, such as, for example, images, clinical data, and cognitive scores. Then we want to see the influence of the cross-validation strategy on how the results can look like. So here we tested just one iteration of a k-fold, then we repeated the k-fold for like 10 iterations, and then you have the repeated holdout with 250 iterations. And so you see that basically whenever possible, and as has been mentioned in the previous talks, it is really recommended to use repeated random splits with a really large number of repetitions to yield like more stable estimates of the performance and better estimates of the empirical variance. Then we wanted to see like how the performance of the linear SVM classifier depends on the size of the training data sets, and so we computed learning curves. Quite unsurprisingly, you can see that when you increase the training set size, then you increase the classification performances. Here, like the balance accuracies. But maybe more interestingly, with the current number of samples available, the points where the results stop improving has actually not been reached. And so this means that more data are necessary to find the top performance of a classifier. And these results really highlight the need for more publicly available datasets on which actually most of the current research in the field relies. So these were like the results for like the features extracted from T1 images, and you can actually find the same results when you use PET-based features. So until now, I've only shown you results that were obtained using the ADNI dataset, but earlier I mentioned also the OASIS and ABLE datasets. And these different datasets actually allow us to see how consistent our results are across datasets. So here I just pick like the cognitively normal and AD subjects because it's the only basically diagnosis that you find in like the 3 datasets. And you can see that, for example, in terms of age, gender balance, it's probably quite similar. It just maybe with a CDR, which is a clinical dementia rating score, the subjects from OASIS tend to have like a milder AD than the other ones, but other than that, the populations are quite similar. And so we can compare the classification performances obtained from the different datasets. And actually, you can see that the highest balance accuracy are obtained for the ADNI datasets, that comprises the largest number of subjects. For ABLE, the balance accuracy is lower and appears like less stable, and this can be seen from like the higher standard deviation. And for OASIS, the balance accuracy is even lower and also has a high standard deviation. And so to be clear on the configuration here, we both trained and tested the classifier on the same datasets. But what we can do also is test how the method generalizes across datasets. And so here we train a classifier on ADNI and we test it on ABLE and OASIS. And like for like comparison, you can also see the results obtained when training and testing on ABLE and when training and testing on OASIS. And what you can see is that when you train on ADNI and test on ABLE or OASIS, the balance accuracy is higher than when you train and test on ABLE or OASIS. And so this basically means that the classifier trained on ADNI, on the ADNI data, generalized well to ABLE and even to OASIS. And the fact that they perform so even better than the classifier trained on ABLE or OASIS, this demonstrates that the increased training size is actually the key factor for improving the performance, because really ADNI has like the most like subjects. So to conclude, so during this presentation, I've described elements that contribute also to make evaluations of machine learning approaches like more reproducible and more objective. So the reproducibility is the ability to reproduce results based on the same data and experimental procedures. And there are actually several key elements to guarantee this reproducible research. And these include, for example, data sharing. And if you cannot share the data, or the data cannot be like redistributed, then it's important to provide tools to automatically convert these public data sets into a community standards. And it's actually more efficient and it saves time when compared to simply making the list of subjects available. It's also important to store like data using community standards for the reasons that I've mentioned, and for also, for example, like it's easier to share data. Then it's also important to fully automatically manipulate your data, and finally to share the code. And I've also like highlighted that an objective like evaluation of a new approach. So for example, when you want to evaluate a new classification algorithm or a feature extraction method, so this requires testing this specific component without changing the others. And this is again possible by using standard really approaches for the preprocessing and feature extraction and standard classification tools to actually set a baseline. And you also need like a rigorous validation that includes a large number of repeated run-up split to extensively assess the viability of the performance. You need to report like the full distribution of accuracies. And actually, like all the violin plots that I've shown you, they were obtained with like NeuroPredict, so which was presented earlier by Pradip. And you also need like an adequate like nested CV to optimize like all your parameters. So I'd like to thank all the people from the team, and we are quite a few here, so if you want to ask questions about clinical or other things, we are here. And thanks for your attention.
Thank you, Ninon.